All right, I'm joined today by a good friend of mine, Sage Arbor, who is a biochemist at Duke University. Sage, thanks for joining us and taking time out of your schedule. I wanted to have you on for today is to teach us a little about biology, the medical and research system works when it comes to a rare disease like Sanfilippo Syndrome or similar, and just teach us a little about the biology. Sage.
Sure thing. Yeah, happy to be here. We're talking about these mucopolysaccharidoses, these diseases. Basically, we're talking about this one thing called GAGs, glucosaminoglycans. And the main thing is that we humans are basically big bags of water. And these GAGs hold they're made of a bunch of sugars and they hold the water. So they're in a bunch of your tissues, and in volume, they're all over the place. But in these diseases, we don't metabolize them correctly. We don't break them down. So they have to go into something called the lysosome degraded. And when that can't happen, they cause all sorts of problems. And depending on the type of MPS syndrome you have, that manifests itself in a different part of the body.
MPS, what does it have that's dangerous, and what's the problem here?
Kind of think of our body as like gelatin, like a big jello. Kind of what those things are. And they get cleaned up. And so as they're getting shuttled around, imagine bringing them into like a recycling center. And the recycling center is the lysosome. And then when they get in there, they can't get recycled. So the recycling center just grows and grows. There's rare cases where the lysosome, the recycling center bursts. But it different bad things happen in different ways. And so MPS3, one of the big issues is it happens in the brain. And so that causes problems, one, to treat because it's very hard to get drugs into the brain. And two, it's a problem because the brain is kind of more fragile and causes problems quicker when things go awry. The main issue is like the blood-brain barrier and getting things in and out of there. So like gene therapy is harder to get in there. It's harder to clean things out of there. This is one of the problems of treating Alzheimer's and those types of diseases as well. But mechanistically, the same things are happening at a cellular level throughout the body.
What are we missing in a kid with Sanfilippo syndrome that an ordinary typical kid does not have? Why is this causing so many problems?
Enzymes are proteins, and people get confused by this because proteins make up our muscle, but proteins also are like these little nanomachines that do things in our body. And our DNA codes for the proteins. Each parent of a Sanfilippo child has two DNA sections that code for these proteins. And so a parent doesn't have Sanfilippo, but their child does. They inherited one copy that's defective and one copy that is functional. That's called heterozygous. And if two parents have that and they both give their child the defective gene, there's a 25% chance of that, then the child has Sanfilippo. And so as you're trying to maximize the human condition, you can target things differently. Preventive, therapy is often the best. So, for example, the cost of sequencing the human genome has dropped precipitously and is now under $1,000. Unfortunately, it's plateaued a little. I can show a graph of that if you want, but it's about $900. Imagine if every person in the U.S. could be sequenced, and then two parents would know, oh, we have a 25% chance of having a child with this. They could pre-screen an embryo. They could choose kind of the child that doesn't have this, and their whole body for the rest of their life wouldn't have that genetic disease, right? That's kind of like preventive. Then there's this realm of you just found out your child has it, and that's where you're like swinging for the fences. We need to do a clinical trial because that takes a really long time. And then there's kind of ladder where clinical trials aren't going to come good enough and you're increasing the quality of time that you have with your child.
In my particular case, we took Connor for genetic testing when he was about 5 years old. We never got back a result that said he had Sanfilippo syndrome. Later, we did. I don't understand why, if they're sort of mapping things out at his geneticist, why did they not catch this at 5?
There are a few reasons of that. The most likely one is there's two general ways of sequencing. One way is called a microchip, and one way is kind of the more classical actual sequencing. In a microchip, what they do is they've pre-printed thousands of genes, DNAs on the chip, and then they flow the sample, your child's DNA across it and see what binds. And they can instantly see, oh, they have this mutation. So the benefit of that is it can be pre-made, it's fast, and it's cheaper. So a lot of people have used that. The problem is if they don't pre-print the exact genetic defect, they miss it. So there's more false negatives. I mean, it looks like the kid's okay, but they're not because they didn't get it on the chip. Now, those chips have gotten a lot better in the last 5 years. The other way is you're actually reading every part of the DNA. And even there, a lot of times, you're almost never are you reading every part of the DNA. So most of our DNA, people used to term junk DNA. It doesn't code for the proteins. So people really just care about, let's sequence the part that codes for proteins. But usually people are not actually having their DNA sequenced, they're having that microchip. Now there's things that can sequence whole genomes again for like $900. Like nanopore is a really kind of novel one that's like funnels your DNA through this molecular machine that reads the DNA like a ticker tape coming out. And you're actually seeing the whole genome. And there's more classical ways of sequencing. But so basically, if you're having your child sequenced, you could ask, is this a chip? Is it a microarray? Or are we sequencing the ATGCs? And that will tell you the answer.
In the case of Sanfilippo syndrome, I recall that there was probably traditional blood tests, and then to confirm, so they had like a 97% level of confidence that he had this, but then they went back and tested enzymes of the two parents, and that I guess confirmed it. But it sounded like they also looked at physical features and other aspects that went into the genetic testing process. Would that be necessary? Because it seems like it shouldn't need that qualitative stuff because it should be so quantitatively determined.
Yeah.
Does that make sense?
That makes sense, and I completely agree with you. So I am not a clinician, I'm a PhD, and I agree with you. It seems like you should just take a cell, sequence the cell. I might have done that for cost reasons at the time, and I don't know the current state of the clinical field on if they're actually sequencing the patients or not. Yeah, it is the case where every cell in the body has that same genetics. There's some diseases like X-linked diseases where some of the cells could actually be different, but this is a case where you can basically take any cell that is from the patient and get the DNA that you need.
If a family has had children with Sanfilippo syndrome, and they know the odds are, roughly a quarter for their next kid if they don't have anything interjected. There's a way for those two parents to prearrange to have a child that does not have Sanfilippo syndrome. Is that what you're saying?
There's two ways to do that. But in both of those cases, you're removing the start of a Sanfilippo child. So they would either do in vitro, where they take the eggs from the mom and the sperm and in a dish start the embryos and then screen the embryos and select for a non-Sanfilippo child. Or they just get pregnant and do pre-screening to see if the fetus is genetically going to have two deficient genes and have Sanfilippo. Now, I have no idea if that's clinically done. I'm just saying that scientifically that's possible, but it's a scientific possibility.
The results of trials in the Sanfilippo world, from everything I've read, have been very kind of mixed to really not very positive. And trials have been stopped due to funding issues and health issues. What is so difficult about Sanfilippo versus other diseases where this same basic genetic therapy or strategy seems to have been a lot more effective? Like,
you look at the other MPSs, the multipolysaccharidoses, a lot of them have present in like the liver or the heart or the bone. Sanfilippo presents in the brain because the deficiency is happening in the brain and causing issues there first. And other MPSs, you can do things like enzyme replacement therapy because the enzymes can go throughout the body and get to the heart and help the heart. But you can't do that because it's very hard to get them to the brain. So it's the blood-brain barrier that's really making that difficult. Basically, if another disease that affects the brain solved this, it would help Sanfilippo because a lot of the problem is the delivery mechanism and getting something like gene therapy into the neuron. right? Orders of magnitude more money to spend on all sorts of dementia, right? Where they're trying to treat that, like Alzheimer's is the well-known one, where they're trying to do gene therapy to help there. So if you can ride on those coattails or like get Congress to put money in to solve that problem, it's like a Trojan horse, right? Here, let's put $100 million into Alzheimer's, specifically viral therapy to treat that. And then you're like, oh yeah, we're going to use that same Trojan horse to cure Sanfilippo.
Sage, there are many types of MPS. I believe maybe 7 or 8, but maybe you could correct me. Could you tell me the difference between these MPSs?
Sure. Let me share my screen. Here are the various MPSs, and I've color coded them to show you kind of the grouping. So I'll show you kind of the enzymes that's deficient. Hunter syndrome is MPS 2, and then here's MPS 3, which is actually a whole group of different diseases. They were called MPS 3 because they manifested the same, but now we know they're genetically different.
I assume that really complicates things, that there are 4 types, because the solution for type A will probably be very different from the solution for type C. Is that correct?
Yeah, they're completely different. So you see how they each have a different protein that's deficient, and so you need to replace a different one.
And does a protein equal enzyme? Are those the same terms?
Yes, an enzyme is a type of protein, but everything we will talk about, they're the same. Yep. All these slides, I can give you links to these. These are all Google slides that I used to teach medical students. So these are about 4 years old now, but they're all freely available online. And then for this talk, because I know you, way back then, I put little YouTubes of me talking over them. So anyone can click and hear my explanation of these if they want to. If you want to kind of see like how things diagnose, we can just focus on the Sanfilippos, but this was kind of deficiencies up here that manifest themselves and which each one has. MPS1 Hurler syndrome is the worst at this intellectual delay compared to the other MPSs. For Sanfilippo, you see their double down arrow, their big negative thing is this intellectual developmental delay, right? They have central nervous system degeneration, sleep disorders. I know some of these can have enzyme replacement therapy, but the MPS 3s cannot because the deficiencies in the brain and the enzyme, we can't get there easily.
Could you tell me the difference between enzyme replacement therapy and gene therapy?
Sure. If you think of the enzyme as a nanomachine, right? Like you have this big gelatin molecule and the enzyme's coming and chopping it up, okay? So it can get degraded, right? Enzyme replacement therapy is injecting that nanomachine into the blood, and that nanomachine goes and chomps it up, and over time it gets degraded and you have to inject it again. So it's something you're continually taking. Viral therapy is you put the DNA in a cell so the cell makes that nanomachine all the time. Viral therapy, gene therapy is a one-time cure. Enzyme replacement therapy is a continual need that you have to do, and you have to get it to the right place.
Okay, now this might be a stupid question, but can't the enzymes reproduce on their own, or is that not the case?
No, they cannot, right? DNA is the instructions on how to do things in our cell, and they instruct, make this enzyme. So all the proteins we have, all the enzymes, even like the proteins in your muscle, all of them, the instructions on how to make them was in the DNA. And then that made the protein. And the proteins turn over time, like they don't last forever. So then you go and make more reading the instructions from the DNA.
Okay. All right, let's keep going with this.
Actually, what's happening? This is your gag, this long thing, and this is it getting chopped up and degraded. And at every point, you're doing something. Here, you're taking off a sulfur. Here, you're taking off a sugar. Here, you're taking off a sulfur. Here, you're taking off an acyl group, which means carbon. Here, you're taking off a sugar. And at every step, there's a different enzyme that does this. So there's a deficiency, you have one of the MPSs. So here's MPS 3, and remember, it's actually 4 different enzymes, here, 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 or here. So here, MPS 3A is taking off a sulfur, MPS 3B is taking off a sugar, and MPS 3D is taking off a sulfur. The point is, they're 4 different nanomachines. You have to fix each one for each disease. Let's say the mom has a 100% deficient enzyme. Sometimes it's chopped in half, whatever. It doesn't work at all. The dad might have one that's deficient, but maybe 30% active. So you can think of the enzyme or a protein as like a folded spaghetti noodle, but because of its shape, it can do things. So it can chomp up the gag. Well, if it's folded a little wrong, it can't chomp it up as well, but it can chomp it up a little. And for enzymes, the general thing is that if you have something that works a little, it's usually good enough. That's why when we have two of them and one of them doesn't work, the mom or dad's fine because the one is enough. You're getting 50% activity. Now, if you take that one and you chop it down to only 10%, you're like, it's close to zero. And then you start having problems, right? Sometimes when you have a partially functional enzyme, you can find compounds that bind the enzyme and make it into a better shape and increases its activity. If that were the case, those rare variants could potentially get better. But it's, you're not bringing it back to 100% normally. So it's kind of like on the edges and it's a subset of the population,
All of these MPSs are essentially storage diseases, from what I understand, where they can't get waste out of the body. Where does the waste go when they are able to break stuff down?
Most of it gets recycled. So you need this stuff in your body and it just gets recycled and reused to attach to things. So all these are chemical moieties that you attach again to other things. It would be like a gelatin and it got super crusty and it's kind of like it no longer has the same function. But if you chopped it all back up into its powder, you could rehydrate it and make it useful again. That's basically, it can get chopped up when the recycling is working well enough, then those constituent things are reused.
Could you tell me the difference between a recessive and a dominant trait?
Having one of them is enough. So like here in an enzyme, if you have one allele, you have two DNA copies that are saying how to make the protein. If one's no good, but the other one is good, the good enzyme is enough so you don't see the phenotype. In the recessive case of a gene, one of them is good enough to get the job done. There's other diseases where a protein may form a matrix. It may, hybridize with itself and form a lattice structure. And in that case, if you have one that's bad and one that's good, the bad one kind of contaminates the good one. So there it's dominant because having one bad one makes them all bad. For almost all these genes, we have two copies, right? Unless it's on like a Y chromosome or something. So when you have two, is one enough to make them bad, or do you need both to be bad? And usually you need both to be bad, but there's some where one is enough. So for Sanfilippo, you need both, and that's why you have a 25% chance of the offspring having it.
Could you tell me a little about how research universities like Duke University, where you work, play into the broader full picture of medical and research industry and how they can help move that process along for rare disease patients and others?
Two avenues that clinical trials will get funded are you can get grants to investigate something and do a clinical trial. Oftentimes it's social though, right? Like Pfizer or Lilly will fund this and use us. And so basically once you have that money invested, you're either, it depends what phase trial you're in. But if you're going all the way, you of course need this massive amount of money, and there's gatekeeping steps at which you report back, were the results positive to continue on. And so, you start off in animal studies, then you get to a small set of humans where you're just looking for harm. You're just looking like, does this hurt people? We don't even care if it does good yet. And then once it doesn't hurt people, you can do it in a larger population and say, are we improving from whatever the gold standard is? So in the Sanfilippo syndrome case, the baseline is very poor. So the improvement would not have to be good, right? So in that way, it seems like you have a easy metric for success in the clinical trial. And then another positive is that it seems to be all or nothing with Sanfilippo, right? Like we know the cause, and so you're likely going to cure it or not cure it. It's a kind of a unique situation compared to many disease states. Many disease states that are very lucrative, that there's a financial incentive to go after, are much more complex, like, obesity caused problems that affect all the organs in different ways, and you're marginally improving something, and the drug that you're using isn't really more effective than like eating less and sleeping. This is a different case where we know the problem, We know the solution. We just technically have to get it done.
Sage, could you tell me about the decision for a research university to get involved in a certain cause versus that cause only being pursued on the commercial side by one of the big pharmaceuticals? Like, how are those decisions made, and is there collaboration between the two sides when handling or trying to solve a complex disease?
There is collaboration, doesn't have to be, and oftentimes there's not. So it's a case by case basis. It often has to do with where the papers were done originally, like who's working in the field. So if an academic in a university is kind of at the forefront of the field, and then pharma finds that they want to go after this, they'll then collaborate because the person of excellence is at a research institution. Whereas this is just generically, like, this is not always the case, but if pharma has been doing that research for decades, or, then they have the expertise in there where they can just run it themselves. So I think it has to do more with where the expertise is. Sometimes it also just has to do with the logistics of, like, okay, this, Duke set up to do a whole bunch of hospital work, and we can use them to quickly do it. But for Sanfilippo, again, because the issue seems to be the mechanistic science of getting gene therapy done, it's still mainly at the science level. It's who's at the top of the field and kind of proving the metrics we need to show that the genes can get where we need them to go. So in short, the university doesn't actually have ownership of the professor at all in the height of their field. So for example, if a professor chooses to leave a university, if they have a grant from the NIH, they take the grant money with them. And they even take any equipment they bought with the grant. So the PI, the principal investigator, the researcher, is really kind of the king of their fiefdom. What the university does own is they get a cut of whatever grant the professor gets for overhead and that sort of thing. And more importantly, to actually get a lot of science done, you have to be at a top tier enough university that you have the support of many people to get things done. So it's not an army of one, right? So you're focused on this one part of science, but then someone else is growing the cells and running the mouse house to do the studies, right? And so there's less failure points, and it's kind of increases the percent of getting across the finish line.